In this presentation, we will explore the extraordinary biological journey that occurs from the moment a sperm cell encounters an ovum, all the way through fetal development and finally childbirth. Pregnancy represents one of the most coordinated, complex, and finely regulated processes in human biology. It involves not only the developing embryo and fetus but also profound physiological adaptations within the mother, adaptations that support growth, maintain the pregnancy, and ultimately prepare both the fetus and the maternal body for labor and delivery. As we move through the stages of conception, implantation, placental development, and parturition, pay close attention to how each step builds upon the previous one. The events of early development set the foundation for a successful pregnancy, and even small disruptions in these early stages can have significant consequences later on. Let's begin our exploration with the very first step, fertilization. Although it is a single sperm cell that ultimately fuses with the egg, human fertilization is truly a collective effort. Millions of sperm are released during ejaculation, yet only a tiny fraction survive the journey through the female reproductive tract. Along the way, they face acidic environments, cervical mucus barriers, and immune defenses that eliminate most of them before they ever reach the fallopian tube. Once the surviving sperm reach the secondary oocyte, they encounter the corona radiata, a thick layer of granulosa cells surrounding the egg. These cells must be forced apart, and this requires the mechanical movement of hundreds of sperm working simultaneously. Beyond this outer layer lies the zona pellucida, a tough, glycoprotein-coated membrane that protects the oocyte from premature fertilization. To break through these protective layers, sperm undergo the acrosome reaction, releasing powerful digestive enzymes that gradually weaken the zona pellucida. This process is not instantaneous. It depends on the cumulative enzymatic activity of many sperm before a single sperm is able to reach the oocyte's membrane. Once a sperm succeeds, it binds to specific receptors on the egg's surface and fuses with the cell membrane. Immediately afterward, the oocyte initiates a rapid cortical reaction that hardens the zona pellucida, preventing any additional sperm from entering. This ensures that fertilization remains monospermic, preserving the correct chromosomal number for normal embryonic development. Following fertilization, the zygote begins a tightly timed process of cell division and development. Approximately 30 hours after conception, the first cleavage division takes place, forming a 2-cell embryo. Subsequent divisions occur roughly every 12 to 15 hours, producing a 4-cell stage, an 8-cell stage, and so on. Importantly, during cleavage, the overall size of the embryo does not increase. Instead, the single-celled zygote is partitioned into progressively smaller cells called blastomeres. By the third day, the embryo reaches the morula stage, a solid ball of cells resembling a mulberry. As it continues its journey down the fallopian tube toward the uterus, fluid begins to accumulate inside the morula, eventually forming a hollow sphere known as a blastocyst. This blastocyst contains two distinct populations of cells. The inner cell mass, located on one side of the blastocyst cavity, will eventually develop into the embryo proper, giving rise to all of the tissues, organs, and systems of the body. The trophoblast, the outer layer of cells, will contribute to the formation of the placenta and the membranes that support pregnancy. Around day 7, the blastocyst reaches the uterine cavity and begins the process of implantation. Trophoblastic cells adhere to the endometrium and begin to invade maternal tissue, anchoring the developing embryo securely within the uterine wall. During this time, the ovary forms a structure called the corpus luteum, which secretes progesterone to maintain the uterine lining and prevent menstruation. All of these early events, cleavage, blastocyst formation, and implantation, lay the groundwork for a successful pregnancy. They set the stage for the next major developmental milestone, the formation of the placenta. The placenta is one of the most critical organs of pregnancy, and remarkably, it begins forming almost immediately after implantation. Even though it belongs to the fetus, the placenta is composed of both fetal and maternal tissues, creating a highly specialized interface that supports the embryo throughout gestation. The early embryo develops several extraembryonic membranes, each with its own unique role. The amnion forms a fluid-filled sac that cushions the embryo and provides mechanical protection. The amniotic fluid also helps regulate temperature and allows the fetus to move freely, which is essential for musculoskeletal development. The yolk sac, although small in humans, plays an important role during the earliest weeks by forming the first blood cells and helping establish the embryo's circulatory system. The allantois contributes to the formation of the umbilical cord and helps organize early blood vessels within the placenta. As the trophoblast expands, it differentiates into two layers, the cytotrophoblast and the syncytiotrophoblast. The outer syncytial layer forms finger-like projections known as chorionic villi, which extend deeply into the maternal endometrium. These villi dramatically increase surface area and allow maternal blood to bathe the fetal capillaries without ever mixing directly. Over the course of the first trimester, the placenta matures into an efficient exchange organ, delivering oxygen and nutrients, removing carbon dioxide and metabolic waste, producing hormones such as progesterone, estrogen, and human placental lactogen, and providing immunological protection by selectively allowing maternal antibodies to cross the placental barrier. The placenta is truly a lifeline, and its proper development is essential for fetal survival. Parturition, the process of childbirth, is a remarkable physiological event that involves synchronized actions by the fetus, the uterus, and the maternal endocrine system. Labor begins when hormonal signals shift the balance from maintaining pregnancy to preparing the uterus for contraction. The rising levels of estrogen toward the end of gestation increase the uterus's sensitivity to oxytocin, while prostaglandins soften and dilate the cervix. In early labor, the fetus moves downward toward the pelvis, and the cervix begins to efface, thinning out, and gradually dilating. Contractions become more coordinated and more forceful, helping the fetus descend further through the birth canal. In many pregnancies, the amniotic sac eventually ruptures, releasing amniotic fluid, an event commonly known as the water breaking. This further increases pressure on the cervix and enhances contractions. During the second stage of labor, powerful rhythmic contractions, driven by a positive feedback cycle of oxytocin release, expel the fetus from the uterus. The maternal body undergoes significant structural changes during this stage, with the pelvis widening and the birth canal stretching to accommodate the passage of the newborn. After the infant is delivered, the process is not yet complete. The uterus continues to contract to detach and expel the placenta, a stage known as the delivery of the afterbirth. These contractions also constrict maternal blood vessels at the placental site, preventing excessive bleeding. Parturition marks the culmination of pregnancy and the beginning of independent life for the newborn. It is a testament to the precise coordination between maternal and fetal physiology.